my pleasure to have Shili Lin today with us. Shili is an alumni from the program. We did back, I won't say comments.
Elizabeth is behind you.
Oh, right. And she was student of the Research of Thompson. And actually, Shili and Eric, student of the Research are here as an interview for Elizabeth to be published later this year. So it is a pleasure to have the two of them here, and in particular to hear Shili speak about her work. She's currently thank you, Shili, for being here, and please.
Thank you. It's great to be back, and thank you for the invitation in the introduction. So yes, I did start my statistical journey here at UW in this department, and I have proof of that. If it works, well, I guess not. So, okay, and this is the proof, right? Okay, so the picture here was taken before people have cell phone with cameras. So it wasn't really a selfie that was just out of focus. It was actually, I had a Nikon camera. I handed it over to one of the, I don't know, a passerby or a fellow student from the department. When this developed, this was how it turns out. The picture was blurry, but memory is not. I still have really fond memory of the department, and I want to thank Elizabeth for introducing me to statistical genetics. I have zero background in biology or genetics. Reading her book on pedigree analysis in human genetics, and her proposal, her NIH grant proposal, they just got funded. I was really fascinated by concepts like genetic isolate, never heard of, quadruple second cousin marriages, recessive diseases, and the fact that statistics have been used in genetic problems. I was hooked and never looked back. Statistical genetics has been really my main area of research to this day. And so today I want to talk about finite mixture model, and I just need to acknowledge my collaborators. So what I'm going to talk about, I joined work with Abbas and Masu at McGill University and Farhad at UNLV. And so I started looking into finite mixture models back in the early 2000s because finite mixture model has really wide applicability in problems where there's heterogeneity. And heterogeneity is a norm in human genetic data rather than the exception. And so my first project into this was looking in locus heterogeneity in linkage analysis. And that was in the situation, so it's in that context, locus heterogeneity is really thinking that in different families, the gene causing the disease may be different. Okay, so in this family, it was the A gene that caused the disease, even though the phenotype are the same, gene may be different. In this family, it was a different genes. And so if you would be indiscriminately including everybody for your linkage analysis to do gene isolation or the locus isolation, then your signal is going to be diluted. So you would only want to include the family that actually being affected by this particular disease gene. If you want to isolate the gene there, the trouble here is that you actually don't know which family is affected by which gene. And so therefore, a mixture model, or called mixture, can be useful in that setting. And our work was motivated by two studies. One is a lung cancer study. The other one is an asthma study, where we were really interested in being able to isolate the disease gene. And so our contribution was basically allowing each family to speak on its own on what is the probability that they're actually contributing to the localization of the disease gene. My next example is actually not in genetics, but it's in biomedical applications, where one is interested in understanding whether speech patterns can be used for monitoring Parkinson's disease progression remotely. You record your speech pattern and it's being monitored by doctors somewhere else. And the reason for doing that is because the standard test, this unified Parkinson's disease rating scale, is actually quite elaborate. It cannot be done anywhere, had to be done in hospitals. It's quite expensive. So for people who live outside of the big city in more rural area, it's both cost and time commitment that you have to go to big. And so therefore, if there were, if the speech pattern, which can be recorded and monitored, that would be the best. And so what happened was that the doctors had already, or researchers had already done a lot of studies and feel like there may be a connection, but there may be heterogeneity there. What we did was we did a mixture modeling and did find out that for people who are in milder situation of the disease, it was 3 speech patterns that may be linked to the standard test. For the people who are more progress in that disease, it's a 6 different patterns that test.
I'm sorry. Your slides are, your screen is broken.
It's frozen. I can't even say stop share. Okay, here we go. Let me try to reshare again. Share. Is it better? Okay, that worked? All right. Okay, so this is a situation where actually we're looking at high dimensional data now because the speech pattern, there are lots of speech patterns that they can quantify. And we not only want to look at the linear pattern, we also want to look at quadratic patterns. So there are quite a number of patterns. And our contribution here is basically a model selection, a way of selecting the key patterns that are affecting, that are influencing the outcome in each of the components. And we're allowing the numbers of covariates to grow with the same size. Want to show you a couple more examples. The next example is actually motivated by a study that tried to understand whether or not DNA methylation, which is types of epigenetic data, can be used for prognosis of ovarian cancer patient after surgery. Because there was belief that DNA methylation plays a role in determining the disease-free time as a way of prognosis. And there were different research in the literature coming up with different conclusions that I will show you later. But one of the main idea was that you're not going to have the same homogeneous patterns in all ovarian cancer patients, in particular, depending on the disease stage when the surgery was happening. And so what we did was that we could not use the Parkinson's disease model because disease-free time is variable subject to right sensory. And so there we have to come up with a different set of statistics for that, but still in the vein of sparse model situation, because the DNA methylation can be a huge number. Even with the K450K array, it's a huge number of DNA methylation that you have to be contained with. Next example they want to show you is making this is where the problem becoming even more complex, and that is you have this regression setting within each of the components. However, the regression coefficient is not a fixed number. It actually can vary over time. And in this example, we can see that the effect size of BMI, this is body mass index, on an outcome variable is actually changing over one's lifetime. And same as the effect of a SNP. And that calls for a much more complex modeling that we would have to deal with. And that is where I'm going to stay for most of my talk. And towards the end, I hope that I will have time to come back to the survival model and give you a little bit a snippet of that as well. All right, so the motivating example that get us to work in this is because of the data set collected by the in Fangchenggang, which is a coastal city in Guangxi province in China. It's crossing the border from Vietnam. And there, the researchers at Guangxi Medical University was interested in knowing whether both environmental genetic factors may have an impact on a particular protein called osteocalcium. So this protein is made by in bones. And people were interested in that because OCA is believed to play a role in metabolic regulation. And in particular, if one has higher OCA level, that they're associated with a better treatment outcome because in osteoporosis, because you increase the bone density. And this was the data set that was studied for quite a while before we got our hang on that, at least for a couple years before we get our hang on that. And people have found genetic variants on chromosome 7 that was linked to OCA, but they have also found effect of age when they were trying to use age as a variable, as a interaction variable. They were finding significant effect, but the result was inconclusive. And there were lots of talk about that age may play a role in an uneven way, not just in a linear regression setting. And this is where we got interested in looking at this problem. And so here, basically, it has all the complications that you could imagine. There's population heterogeneity, because not everybody have the same relationship. And so therefore, it calls for really using mixture modeling. And there are a large number of genetic factors and non-genetic. And not all of them has a role in affecting the OCA. And so therefore, you have to do a variable selection because there's sparsity there. And again, what we got interested was really the third point, which is that the effect may be age dependent. Depending on someone's age, it may not have the same effect. And so that really got us interested in this varying coefficient model. And not only that, the probability that one belongs to a component may be changing over time as well, depending on their age. And so therefore, we are also interested in looking at these varying mixing proportions as well. And so with that, I want to give you a little bit more dissection into the model. Oh, before that, okay, I just want to show you one. So we have heard that the effect is age dependent, but we want to see it for ourselves. So we said that, okay, so we believe that there is a heterogeneity, but how about we simply fit for a mixture of regression model, see what happens, and these are the residual plot. Doesn't look that great, right? Especially for the second one, there's a curvature there, so they're indicating potentially there could be an age effect. And if you see that 0 is there, right? So it's like pretty uneven. Okay, so this is where we said that, okay, so what we want to do is that we really want to look at maybe come up with a method that can complete all these problem here. And so now it's time to give you a little bit more into all these different components and put that together with some notations. Okay, so we say that population heterogeneity because there may not be a single population, there are subpopulations. The trouble here is how many subpopulation here is actually unknown. You don't know how many, and this is a parameter that you have to estimate. An individual belong to one of the subcomponents, the subcomponent can change over time depending on the age. And so therefore, that part is also varying according to what we call an index variable. So this is a single variable that dictating how things moves dynamically. So we have these regression settings, right? Within each component, you want to look at the relationship between the covariates and the outcome variables, but it's also changing over time, depending on this index variable value. And so putting all together into a model is the following. So we have our density function expressed as a mixture of multiple components, where C is number of components that we hypothesize, which we actually don't know. And all the function of interest or parameter of interest are all indexed by this variable E. Now, this is our mixing proportions, and that can change according to the index variable E. Our density function is assumed to be coming from a parametric family that we know. And we have been looking at either Poisson model or binomial or normal distributions. And it basically is characterized by two functions here. One is the mean and one is the over dispersion. The mean is basically related to the nonlinear predictors through the link function the usual way. And so, so this model will be able to accommodate the features that we are talking about here, that all the 4 features that we want will be able to be accommodated in this model. And so you may say, okay, the only thing that this model would not do, although it accommodates all the other things, is that it actually doesn't say a sparsity. Sparsity ought to be inferred. You need to do your statistical inference to get at the sparsity. And it's actually critically important because in finite mixture model, the numbers of parameters that you have to estimate can grow quite big if the numbers of components is large. And in FMBCR, in what we're doing, these are not just fixed parameters that need to be estimated. These are fixed functions that have to be estimated. It's easily, even if you control the dimension, easy getting to hundreds of these things with the hundreds of sample size of hundreds or even thousands. It's very difficult to work on this problem. So you certainly have to induce sparsity if you believe sparsity is there, which is true for a lot of these high dimensional problems. All right, so let me try to define sparsity in a little bit more precise way. So when we say sparsity in what we are doing here, we're basically saying that there is a set of regression coefficients, which just simply is simply equal to 0, does not affecting things. So the expected value of the norm of the function in the entire parameter space, our index parameter space, is equal to 0, basically. And this is a pretty stringent way of thinking about sparsity. There are plenty of papers in the literature which use a weaker way of defining sparsity, and that's expectation to be less than a threshold rather than equal to 0. But for what we are doing, if we would make it to be just a threshold with the finite mixture, it's becoming too difficult. And so therefore, we use this more stringent definition of sparsity, just assume that's equal to 0. And that's coming to play when we start talking about oracle properties. And so if indeed this is the kind of sparsity that we have in the model, then our parameter function is going to be tremendously by looking at only D0 instead of D covariance. And so what are available in the literature that we can draw on so that we don't have to recreate the wheel, so to speak, if we don't have to. And so if one were just looking at finite mixture models, there are plenty of methods available for variable selection, including what I just talked about, the Parkinson's disease one, that is a variable selection in finite mixture regression models. If you simply just looking at varying coefficient models without having the heterogeneity being introduced so that you get these components, then there are plenty of methods there for that as well. However, what we're interested in is putting these 2 things together. And for that, we just didn't find things that can do both. There were methods that, what did I do? Okay, here we go. So there were methods that were focusing on simply do a maximum likelihood estimator without introducing sparsity. But what we want is also have sparsity plays a role as well. And so therefore we have to try to come up with something else. And so what we try to come up with, again, is basically simultaneous inference of estimation and variable selection. So let me just take it apart to make it easier to digest. So let me just say that, okay, let's think about the estimation component. So here now, it's just not the model anymore, we have data. So let's suppose that we have n observations. So these are our triplet of observations. We have the index value in the index space. We have our covariates and we have outcome variables. And we want to use the FMBCR model that I have just tell you about. We assuming that our observations are independent, so you can write down the log likelihood very easily. The trouble is the inference, not writing down the likelihood. And so just to remind you, psi is denoting the collection of all my unknown non-parametric functions. So this is an intractable problem because it's a infinite dimensional situation here. You're looking at functions instead of parameters. And what we decided to do for estimation is that, well, that's we use local kernel methods where we can approximate the function at each position in the local region by a constant. And so that way you can make your infinite dimension into a finite dimensional problem. Okay. And so, so essentially what we did was that we said that, okay, for any values in our index parameter space, we're just going to write down now the local kernel log likelihood by using a kernel function, and H here is denoting the bandwidth. So this is the standard way of doing kernel approximation. And so now, as I said, what we have is locally, we got these parameter vectors. They are no longer functions. And by doing that, it makes it easy for us to devise an EM algorithm to estimate it, because this is becoming a typical finite mixture regression model that can be done. Now, one of you might say, why don't you use some other ways of doing your approximation, right? There are these, say, local or local polynomial or spline, right? Those were very popular. Our goal is to not only being able to model it, but being able to implement it and make it work. And so this is where we do the local kernel to make the EM, to make it possible to do the EM.
This is great. What is the with the beta?
Oh, so yeah, so thank you. Yeah, so beta is the nonlinear predictor function that now being estimated by being approximated by a constant and is linked to the mean. So let me back up to the slides where we have that. One more, two more here. Okay, so that data is our mean, and the mean function is related to your typical regression coefficient, except now it's a regression coefficient function. And so that's just your.
Like the same as a single index.
It's well, I don't quite know whether or not that's the same, but what happened is that you think about your regression, you have X beta, right? But the beta is now has an index there that is looking at the whole space as well. And then it's linked by this link function there, because if we don't have, if we have, say, signal data or count data, right? We need a link function in order to make it to work. But thank you for the question. Where were we? All right. Yeah. So our purpose for doing that was really just for to make it easier. Okay. Where were I? Local signal estimating. Yeah. Okay. So this is where I am. But so far we still haven't done sparsity, right? We just talked about estimating no sparsity, and you would say that, oh, why don't you just do what you did with your parking by introduce penalty function, and there you go. Okay, the issue here is that what we want is a thing called the uniform sparsity. Go back to review this, the sparsity definition here. What we would like is that if a covariance has no effect on outcome, it has to have no effect in the entire parameter space of the index. We don't want it to be in one component, it's this one, and then this is yes, and this is no, right? We want it to be no, have no influence. It doesn't have any influence on that. And so because of that, we can't just do what we have done before to introduce sparsity. We need to do something else so that we have this uniform sparsity. And so what we did was we are going to discretize our index space. So we have n observations. And this n observation is on the space that you are interested in, your index space that you're interested in. So they naturally discretize your space. And so if we look at each one of these and put them together, so if you recall, this was my local kernel likelihood before, but now I'm looking at every single one of them at the parameter value, so at the index value that it is stratified here. And so therefore, I got my total local kernel log likelihood here. And what's interesting here is that now with this, what we are having is a parameter space is finite dimensional and can be put into a matrix form where the numbers of rows are the numbers of observations. For each row is each observation. The local constant parameter that it has is the same number as in a typical finite mixture of regression model. And then make it, I would not say super easy, but they can make it a lot easier to implement EM algorithm for this. Now, in order to do all this, this is where we need to do, right? The typical way is introducing a penalty. Now, how do we introduce the penalty so that we have uniform sparsity? Well, it's all in this group penalty. So in this group penalty, we're penaltying all the parameters within each of the index space the same within its component. And because of that, what we're going to achieve is uniform sparsity. Either a covariance has an effect or it doesn't have an effect in the entire index space. And I'm going to show you, that's what I'm going to show you next. Okay. And
So the penalties, well, what is the penalty?
okay, yeah. So we actually looked at all the common penalties that we use. We look at lasso, adaptive lasso, SCAT, MCP, SICA. We're just looking at all possible penalties there. And that's yeah. But it's in a group setting. So the P is those penalties, but this is the group setting essentially.
Then which one is the best?
You just have to wait. Sorry. Yeah, you just have to wait. All right. So okay. So just to remind you that we partition our parameter space the same way as what I show you in the sparsity slide. I'm not going back there. So basically, essentially, that's a part of the parameter that have zero inference on outcome, and they're putting into the second part here. So these are exactly zero, and so they make it easy for us to express the oracle estimator, which is basically saying that I'm going to fit the model, assuming those are not even in my model. I'm going to get the estimator, and those are my oracle. My estimator based on the simultaneous inference will be denoted by the psi here, psi hat here, and then there are two components because we did not have prior knowledge that it's supposed to be zero, and so therefore this is what we have. And so indeed, this is what we want, and we achieve that, and this uniform sparsity. The estimate in the entire index space will be zero with probability going to 1. And then it's going to approach what our estimator without assuming the oracle, without assuming the zero, going to achieve approach the oracle estimators. And the most useful one that we found was the asymptotic normality that we could use for our statistical inference. And so with that, I think I'm ready to show you the example and also talk about which one worked out better in terms of the penalty, but you have a question.
Yeah. For the oracle property, basically the rate of conversion is faster than the group N, if I understand correctly by the location here.
It's the same. It's big O, not small O. That's a big O, and that's a coefficient that I ignore. So if you read the paper have all these complex coefficients that are all constant, not with anything to do with N. So I just simplified. But it's a big O, not a small O. Sorry, did it look like small to you? Yeah. Oh, it looks. Okay, so sorry, that's a big O. Big O. It's a yeah. All right. Okay, so with that, I'm ready to go back to our example. All right, so the data that we got had initially had 2,100 individuals, somewhere around there. It's a rounding up or rounding down. It's I can't remember. And there were again rounding about 65,000 SNPs, and there were 22 non-genetic covariates. And obviously, we could not with this many people, and with like trying to fix mixture model. Obviously, we could not do this, and none of them are not useful anyway. So we do an initial screening, and with initial screening, basically delete missing data for those which have no variation in the whole population. We got rid of them. If they have nothing to do with the outcome variable, we also got rid of them. And we eventually got down to 43 covariates, which including 36 SNPs and 7 non-genetic variables. But that still is a large non-parametric function to think about if you think about fitting a model with 4 components. And so what we did was that we used all these different penalties that I just talked about, right? Or didn't talk about, but mentioned with the questions. And what it turned out is that scat penalty fit the model the best in the sense that it's very stable. When you use, for example, lasso, sometimes it fits pretty well, sometimes it fits more poorly. Same as some of the other penalty that we looked at. Adaptive lasso was quite good for most of the cases, and sometimes, ooh, it's just like going way bad. And so therefore, we use that because that turned out to be very stable. And it's, we look at up to 4 components. And the best one selected based on BIC was 2 components. All right, so in the first figure that I'm showing here, these are simply the density plot for the 2 components. And so the first component here is what we call the high OCN component. These are the individuals that have high osteocalcium. This component captures those which have low osteocalcium. And we can see that the missing probability for the first component, and we only need to look at the first component because they are complementary, they sum to 1. And so for the first component, at a younger age, the probability belong to the high OCN component is about 65% at the age of 20. And they're going to reduce about 40% by the age of 40. And they seem to be biologically quite interpretable in that. And what's interesting are these 2 nonlinear effects of the covariate. So I just showed 2 as examples. The first one is BMI. It's the same example that I just showed earlier. And that is, we see that for people who have a higher BMI, the body mass index, it's going to have a detrimental effect on the osteocalcium levels because these are all negative. They have negative effect on that. But as they get older, by the time 40, 50, these are really doesn't matter anymore. Okay. So that's what we found. The SNP one is the most interesting. So it has a deleterious effect. That if someone has this particular SNP at a younger age also, it has a negative impact on the osteocalcium level. So it's really interesting. I want to actually take a closer look at this particular one. And so the reason I want to do that is because there were literature supports. So this particular SNP has been implicated in the literature and shows that it really impacting the OCN levels. And so this is why we want to pay attention to that. And in particular, we want to show that in young people that has a bigger impact. But what we really would like to do from a statistical point of view is at least try to substantiate whether or not the non-constant effect, the negative effect are real or not real, although we cannot say for sure, but at least we need to do our due diligence. And so the first thing that we did was check the residual plot, right? After we now including the varying coefficient component. Before we did that with the simply the finite mixture model, regression model, but now we have the finite mixture of varying coefficient model. And this is on the right-hand side is the FMBCR model, on the left-hand side, the FMR. I could not say anything about the first one, but at least for the second component, the residual, at least for me, the residual is better by including the varying component, the varying coefficient model, the varying coefficient in the model, okay, compared to the one with the simple FMR. So that's the first one that we check. The second thing that we check is that we want to know whether or not, because in the previous slide, you just saw the point estimate, and we want to look at confidence estimate to see whether or not the confidence interval indeed equals 0 at younger age. And this is what we have look at the top. Okay, so this gives us confidence estimate. And so at younger age, the confidence estimate does speak to not including 0, so that's a negative impact on OCN. But as one gets older, all bets are off. It's just like very large variability. It's really not covering. It's covering the zero. Okay. The last thing that we looked at is to address whether or not it has a non-constant coefficient by looking at the derivatives of the coefficient of the regression coefficient function. And we did that by using approximation. And what we could see here is that it will, it's unlikely this is equal to zero, right? By just doing this approximation there. So they're indicating that the effect of this particular SNP on OCN is unlikely to be constant over the entire age span. Okay, so with that, I think I'm going to move on to, question?
Yeah.
Okay.
Very good question, but it's interesting that which means that maybe just a piecewise linear function would be
Too, because in the end, what we did was a piecewise approximation, right? So we do the local kernel, piecewise kernels. Possibly that could be could be it, but the we did that was more piecewise linear, right? By using the local kernel, yes. And there was another question.
What kinds of derivative estimator you're using to estimate the derivative? Yes.
All we did was like using the estimate and then using the MATLAB to approximate the derivatives.
Like,
Yeah. That's all we did. Okay, to approximate the derivatives.
Be like a regression out of that and again, possibly a smoother derivative, I guess.
Well, I mean that wasn't the goal, right? Our goal is just try to say that okay, now we got these non-constant functions. Now let's just like try to find evidence. But yes, I mean you could do that to make it smoother, but that wasn't really our goal. Yeah.
Because I'm thinking whether you can also have confidence interval for that, because now you can see some of them are big, away from zero, but I don't know whether it is statistically significant that is big above zero or lower than zero. So I guess would that be possible to have like a confidence interval?
For this,
Yeah.
I have to think about that, right? Because I have to think about that. Yeah, so if these are covering zero, that we're just meaning that it's piecewise constant, and that get back to Abel's questions earlier, right? Yeah. But that's an interesting question. Yeah, interesting question there. All right. So any more questions before I because I know I still need to leave some time for questions, the usual thing. So I just want to quickly show you some results for the other ones. Okay. All right then. Okay. So as I already briefly mentioned, the motivation example was the desire to look at whether or not DNA methylation could be used to for prognosis, and in particular, just looking at its association with disease free time, which is a variable that is censored, right censored. And the reason that we're interested in this was because there were all sorts of results in the literature that caught our attention. And so the first thing was that people were using the TCGA data. This is the cancer genome atlas data, as well as other data sets. And there were really different results coming out from that. One paper would say neither methylation nor mutation of BRCA1 gene was associated with prognosis. Another paper would say the opposite. BRCA1 is a significant factor for improving overall survival based on meta-analysis. Yet another paper would conclude that BRCA1 may have different methylation profile at different stages or in different subtypes. And the last one was what caught our interest, because I think that it tells us that there may be potential existence of a heterogeneous relationship. And because of that, you are getting inconsistent results, and we want to do our own study. All right, so again, this is a right-censored model, and so therefore the usual survival analysis setup where you are observing the T may or may not be the actual time of disease-free time, because it may be censored because people are still disease-free by the time that that it got censored, the study ends, for example. And so our model is the usual survival model where if someone has an observed disease-free time, actually observed disease-free time, it's simply just the density function that is there in our model. Otherwise, it is the survival function that one use. For that, same kind of setting that we're going to assume that there is a mean parameter as well as over this dispersion parameter. And because methylation is a very high dimensional situation, we're going to induce sparsity because in reality, you're not going to believe everything has an effect on the outcome. And for critical reasons, that need to be done. Okay. And so, so what are available in the literature? Well, I already talked about FMR. If you don't have survival, if you are not looking at data that are subject to censoring, there are lots of methods. If you simply look at survival model without assuming their heterogeneity, there are also lots of different methods that I listed here, from the more classical method, variable selection, backward selection, to varying coefficient hazard model, to Cox and accelerated ferry time model. So there are lots of things out there. Now, if you ask, what happens if you put the two things together? Well, we could find one method here, which is the first one. It's a two-step approach. The first step is using BIC to pick out how many components that you have, and then based on that, you use your backward elimination to get your spot. The trouble is that by reading the paper, you know that these mixture order, the numbers of mixture components can be overestimated. And so therefore, we said that, well, okay, so now let's try to see what happens if we actually put them together into the single framework, very similar as what we have just talked about. Now, I'm not going to go into the detail because, again, I want to make sure that we have time for some discussions. And so I'm just going to say that the method that we propose also have the property that we want to have. It has this consistency, sparsity, and asymptotic normality, very much like the one that I went through in detail. So I'm going to skip that. What I'm going to show you are the results that we have found. Okay, so a little bit more about the data sets. And so we're looking at the cancer genome egg loss data set that has 396 patients that has disease-free time after surgery. 28% of them are censored. Okay, so that's not an insignificant amount of non-censored data. We tried this also without censoring, just like whatever it is, we call it true, and we got biased estimate, as you can imagine. And there were methylation level for about 9,000 genes in the promoter region. And what we did was, again, we did screening. We screened down to only 20 genes, which include the BRCA1 and BRCA2 genes that we studied. Same idea, we're going to apply this model with penalty. And we look at both the log normal AFT model and the wild AFT model. And all the penalty that I just spoke to for the other one, we also consider, and surprise, SCAD turned out to be, again, performing better than the other ones. That it also selected two components based on the BIC criteria. And so our result basically spoke to, well, there's indeed heterogeneity, heterogeneous relationship. So these two components indeed more or less correspond to what it was said in the literature, the last example, that the different components really represent disease severity, but we also stratify according to living status, tumor stages, and responsiveness to treatment. So let me show you the results. So in the first one that we have here, all these four figures are the y-axis is the probability of belonging to component one. And we see that the people whose disease has progressed is more likely to belong to component one. The people who has deceased are more likely to belong to component one. The people who had a higher tumor stage is more likely to belong to component one. The people who are resistant to chemo treatment, it's also likely to belong to component one. So that really speak to that component one seem to be capture more of the more severe form of the disease in different ways. The second thing is that, so remember that I show you these three different things in the literature, right? They talk about BRCA1 gene. So we're interested in looking at BRCA1 gene. So the theory is that if a gene promoter is hypermethylated, meaning have higher methylation level, then they're going to downregulate your gene. You're going to have lower gene expression. So there should be a negative relationship between gene expression and we happen to have gene expression for BRCA1 gene, as well as several other genes that I'm not going to show here. It's in the paper. I just show one here because this was the one that kept mentioned. And what we see here is that, okay, so component one are the ones in red squares. Component ones are the one in blue, but have two different symbols, the circle as well as the triangle. So component one basically says that yes, indeed, there's a negative relationship, straightforward saying that yes, there's a negative relationship between gene expression and DNA methylation, even after multiple comparative correction, because as I said, we have 8 genes that have expression level and we correct for that multiple comparison. The p-value was very small. Now, and the negative relationship is not linear. So if you would do a linear look at say the Pearson correlation, you're not going to get that, but we're looking at Kendall correlation and we found a significant negative relationship. Component 2 is a little bit more interesting. If you look at the whole set, there isn't a negative relationship. But if you using tau path, which is basically looked at where it stopped to find the correlation, you found a negative correlation. Component 2 as well. And this may spoke to when people said there wasn't relationship. If they're simply using, look at the entire set of things, you're not going to find it either. So with that, I'm just going to conclude my talk. All right, so I hope that I have convinced you, or at least plain to see that finite mixture model has a wide applicability in statistical genetics and genomics, and more generally biomedical applications, and in particular, finite mixture of sparse survival model and finite mixture of sparse variant coefficient models are relevant for looking at these high-dimensional genomic data when there is a complex relationship, such as hidden underlying population that you try to unearth to understand. And again, I apologize that I didn't have time to go into detail of each of these and show you the simulation study, but we did do extensive simulation studies of investigating all these different penalty functions. And it turns out that even though for the real data, SCAT was winning every single time, right, for the two application, when we did our simulation, none uniformly outperform the others. And so our recommendation and what we have done ourselves was that we look at all of them and we run them multiple times to look for consistency or replicability. Are we getting consistent same results, more or less the same result when we run it multiple times with different starting value, et cetera, right? Okay, and different tuning parameter selection, et cetera. And so, and then we're going to also evaluate based on the selection criteria. So far, we have been using BIC. We tried EBIC, that didn't really work. You know, that's for high dimensional data. They didn't really work, so we just stick to BIC. And so this is what we have done. Run multiple of them, try to look for consistency, and then using a selection criteria. And with that, I just want to thank, grateful to Professor Zhengnang Mo at Guangxi Medical University. He was the one got me interested in his data when I visited Shanghai. He was also visiting, and this is where I got to work on this problem. If anybody's interested in, this was the 4 paper that I touched on today. And thank you for your attention. I can take questions now.
Okay. Last slide. And so here, I guess the open event there about
Sika? So sitcom is sort of like a smoothing type of method. So remember that scat, right? It's a smoothing, the smooth clip methods, right? And sitcom is in the similar vein. Yeah, so that is certainly much less used, right? I mean, MCP has been like, people like MCP a lot, right? And MCP, we also got, I mean, the result, seriously, the result other than Lasso, okay, Lasso didn't seem to be work as well as the rest, right? The rest were all very similar, especially adaptive Lasso, SCAT, and MCP were all very, very similar. Yeah, but we just found that in our real data, SCAT is a little bit more stable. The results were more stable. And that's why we, for consistency reason, we just select that.
Yeah, I think that always more bad among others, although they look similar, but this one seems to be better. Yeah, it's just magical. I don't know, like I previously I thought there was some intuition is because scale probably like they have some order, or probably they can try to recover the actual signal is big enough, they can actually try to recover the like those active set for the coefficient. But it seems just magically perfect. They always work. Yeah.
Yeah, I mean, like as I said, right, you know, it's somehow applications, SCAT seem to work, right? When we did simulations, it's sort of all over the place, right? Sometimes this is better, sometimes it's better, but in application. At one point, we were thinking that adaptive lasso worked the best, right? Because it can be really good, but then in one situation, it really dropped, and like worry about the consistency of the results. And so we decided to just go with SCAT, because SCAT also does well for our BIC criterion values as well. Yeah.
I don't know if you could go back to the first example.
First
the
example.
Time varying coefficients. So I guess one.
The results?
Yeah.
Okay,
So I
here.
guess
Okay.
one of the questions is, so are the observations over time like regular grid, or is it an irregular?
So these are all actually sectional observations. So it's not that you follow them over time. And so these are people who have, it's the male inquiry age from 20 to 80. They take a collection of the sample and did that. And what we are doing, say, time varying, is thinking about that if someone is at an older age, what is their effect on the outcome compared to someone at age. So it's not a longitudinal measurement.
So you're not following the same subject?
No, we are not following the same subject. These are all different subjects. And this is why we could write down our likelihood very easily, because these are independent observations assumed to be coming from the same population.
So then the question is, biologically, if you have an age-dependent effect with a SNP, where it's not a total study, it's a causation study, what does it mean? I mean, a SNP is a SNP, it's not totally a something.
Right. So I guess we are thinking about like a surrogate, right? So you're not thinking about that, this person, that this is like a study where you follow people in time, right? And then this is follow them for their lifetime, because we're from a very young age to very old age, right? So from 20 to 80, basically, and think about, what's their effect, right? So here with
Different environment.
That's very true. And this is why the covariate was also being, other non-genetic covariate was also predict. For example, I only talk about the BMI, but we also look at the cigarette smoking status. We look at the alcohol, we look at exercise, we look at other things as well, try to correct for those things. I know it's not a perfect way of thinking about interpreting the result, but it's with whatever data that we have, try to see whether or not SNP affecting the outcome at different age differently, by also taking into account of all the other factors that they have collected. So there were 22 non-genetic factors that they had collected. They would use them all in our model, and they just like drop out without screening. Yeah, 2-7 only. Yeah. So that's a really good point. So I don't want to over the result. This is just like, using the data that we have, right? So these are just like the sectional study that, what we have is a snapshot of what happened there. in that, yes, very good point.
And I guess, I mean, I am a little bit surprised that the profile really, you have kind of, yes, an increasing trend, but then you have that little drop there. So there is kind of this very
Yeah.
sharp edges
So-
to the estimate.
So we are not really trusting that because it's the edge effect, right? So that's known for any of these types of penalties, right? Models that you take the edge effect, just like when you do spline, right?
Yeah,
Your
but-
spline model, the N is not really very trustworthy.
So it's an edge effect. So I guess that's the part that the part I was trying to understand. So you have, oh, I see. So in time it's a step function or, you know.
Yeah.
Okay,
yeah,
and so you're
yeah.
just falling somewhere.
Exactly, yes, exactly. You're falling somewhere in your step. Exactly, then close to the edge, we're not really trusting that. And this is why I say that by the time we get to 40, 50, it's more or less meaningless, right? Because it have this like, I mean, nobody's going to believe this, right? I'm not believing that myself.
We have one here or? No.
Yeah.
Yeah, no, that's just didn't seem to to have
Yeah.
make it higher order. Yeah, just like your spline, right? That you correct for the boundary, right? And then you can make it like so that your boundary is more trustworthy than that. But no, yeah, but again, it's difficult to think about in what we want to do, right? Because we also have to think about practicality. We want to make it possible.